We're all here because we are interested in the deep sea, or we already love the deep sea, or we want to get to know a little bit more about the deep sea. This is a space to be curious and to learn and to get to know each other. The people that are here is because we're all in it together. We're all interested in the wonderful environment and creatures that is the deep sea. And so I encourage everybody, if you have any questions at any point, feel free to raise your hand or use the chat function. If you don't feel comfortable speaking up, but would like to have your question, I'm more than happy to ask the question for you if that sounds a little bit better for you. And we want to thank our two speakers today, Paola and Meghan, for being here today. And so without further ado, I will give the floor to Miss Paola.
Awesome. Thank you, Frida. Hi, everyone. My name is Paola. I am a second-year PhD student at the University of California in Santa Barbara. I study the deep sea and any remote marine places. So our lab is called the Cold Dark Ventus Lab, and we study both the deep sea, which we're going to talk a lot about today, and Antarctica. So more than happy to expand on that as well. And yeah, I'm from Puerto Rico. And as you know, like if you're near the Caribbean or know a bit of geography, you know that's right where hurricanes love to pass. So my background is actually in coral restoration and coral reef research, and they are one of the most amazing natural barriers that we have against these big waves that hurricanes create. So that's how I got involved in this field in the first place. And I love to nerd out later about deep sea coral reefs and Meghan's background, the Lamelliverkia worms and those deep sea ecosystems, because they really have more in common with stuff we see when we're snorkeling than we think. Yeah, happy to be here. And
my name is Meghan Jones. I'm trying to breathe the butterflies out right now, friends. I'm feeling it. I'm an artist. I'm an author. I primarily work with images from the deep ocean, usually lower than 1,000 meters. What else? Oh, I do a little bit of work for the Deep Sea podcast as a production assistant, and I'm just really happy to be here and share my art with you and nerd out a little bit because this stuff is pretty rad.
Awesome. So Frida, I think we're going to start a bit with the science first.
Yes, please.
Awesome. Okay, so I think the description was great, but the idea of this is that we're going to have like someone speak about the science and then someone speak about the art. So I will start out with the deep sea, right? Like when we talk about the deep sea, it's pretty much like, what is the deep sea? And when we think about it, it's more than 90% of our oceans are actually considered the deep sea when it comes to the surface ocean. Like most of them have more than 200 meters below them of ocean water. So it's pretty much a giant portion of our oceans, right? And of it, seen to human eyes, is less than 0.001%. So very, very low. And so it's pretty much still unexplored. And I live by the philosophy that's definitely someone else said and not me, but we can't protect what we don't know. And right now, the deep sea, and we're going to talk a bit more about this later and another brief chat, but there are a lot of threats around it right now. So yeah, when it comes to the deep sea, it's great to know first what we're talking about. So when we look at the water column, the first 200 meters, we call that the photic zone, because basically photic means light and light goes through there. So there are organisms that depend on light, so they can flourish there. But beyond that point, there's pretty much darkness. So the deep sea is characterized by it's very cold, it's very dark, there's a lot of pressure. And that's why our lab is actually called the Cold Dark Ventus Lab. We study what's on the seafloor, and then anything that's cold and dark, we're there. And just this becomes like a very interesting place because there's a lot of endemism, and just basically a lot of organisms that live in the deep sea can only live in the deep sea. And what's truly amazing is that there's so much left to explore, right? And so they have all these crazy type of evolutionary features that I think Meghan depicts so well on her paintings. So I'm really excited for her to show you what she has. And this is just the tip of the iceberg what we're exploring. So there's a lot going on the deep sea. And since it's dark, these ecosystems also don't depend on light, which is crazy, right? When we think about a food web and in land, we think about the primary producer. So we have plants, and then something eats the plants, and then something eats the thing that eats the plant. And then we have like a chain going on. We have a whole food web. But on the deep sea, we don't have plants. We don't have light for plants to do photosynthesis. Instead, we have these crazy organisms and these microorganisms that we can see with our own eyes, but we know they are there. And they use these chemicals and these nutrients that go around the deep sea and produce energy out of them. And we have these whole food webs based on chemiosynthesis, which instead of having photosynthesis, we have this other side going on with chemicals, especially around carbon. So carbon is a big deal when it comes to the deep sea. It's one of the main energy budgets, we can call it. And yeah, around that, there's also, so when we think about the deep sea, because we can't see it for the longest time, most people didn't think twice about what kind of life was there. We just know it was weird. We don't know what's going on there. Maybe it's just like a desert. And the more we explore, the more we know that there are a whole range of different type of entire ecosystems that can go miles and miles. Like, for example, the deep sea coral reefs. Like, we have something called, it has changed like 5 different names in the last 5 years, but it's a type of hard coral. So when we think of coral reefs, we have the exact same thing, but on the deep sea. And Lophelia pertusa in 2019 was the name. It has changed right now. I don't remember honestly what it is, but it is miles and miles of hard substrate. And you have the entire function, like the ecosystem service of a coral reef, but a 2,000-meter step. So in the end, it is just highlights of how much we don't know about these ecosystems. And that's only one type of ecosystem that we can define. We also have hydrothermal vents, methane seeps, like Meghan's background. I really hope she talks about it. It's amazing. Those are called lamellobrichia worms. And those ecosystems, those worms, they grow larger than me. And for example, I'm like 5 feet, 5'2" tall. That's crazy. And these worms, in the deep sea, there's something called gigantism. Hopefully I'm translating this right, but everything just goes so slow and so large. So they tend to be very resilient, but at the same time, it just highlights how rare they can be. It takes a long time to create these ecosystems in the first place. So when we think about potentially harvesting these ecosystems, we need to think about, well, how much time did it take in the first place for this ecosystem to form? And if we try to extract something from it, how much time is going to take to get this back? If we can make this ecosystem go back to its original state in the first place, and even more of a question would be, well, do we know how it looked like in the first place? We already extracted some of it. So those are some questions to think about as we, in the potential future reef chat. I know there might be a potential deep sea mining reef chat and stuff like that. So yeah, happy to answer any questions about the deep sea. There's still a lot to talk about, but I would love to pass it on to Meghan and her work, because we can't protect what we don't know, and this takes a whole village, right? And one of the most powerful tools we have, especially science only can get us so far. We need the whole community, and we need to share what we do. So Meghan, would love to know more about your art.
Okay. I need the power to put the PowerPoint on.
You've got it.
Yeah, baby. Okay. Am I sharing my screen? Can y'all see what's going on over here?
Not yet.
No.
Oh. Well then, let's see here. Yeah, I know. Uh-oh. Well, if anybody wants to walk me through how to share my screen, that would be amazing.
Of course. On the bottom of your screen, there's going to be a green up arrow.
Yep.
If you
Oh,
click
yeah.
that
Wonderful. Perfect. All right. All right, we got it. We got there in the end, right? Perfect. Let's see this as a slideshow. There we go. Can everybody see it? Yay. Okay, super duper. This is my opening screen. It has my name on it, my socials, all the QR codes. My name is Meghan Jones. I am an artist and an author, which I mentioned previously. I have been an artist my whole life, in various forms. I graduated with a Bachelor of Fine Arts, actually out of the Fibers and Textiles department. And I spent years designing knitting patterns, actually, for magazines like Vogue Knitting magazine. So I have a huge body of work in that realm. And then about 2 years ago, I listened to a podcast. I listened to the Deep Sea podcast, and it was fascinating. I joined their Discord, and on their Discord server, these are the original pictures that I saw. So you can see in the bottom left-hand corner here, this is the original photos that Professor Ellen Jamieson shared on our Discord channel. And you can see my little exploding head. I was the first one to post an exploding head emoji because, man, this blew my mind. I had no idea that these creatures lived down here. I had no idea how beautiful they were, how fascinating they were. And it just really captured my imagination. And so I wanted to just try painting one. So I was just, Alan was on there one day and I said, hey, like, what does it take? What does it take to get permission to paint from these photos? And he's like, man, anything you want. Like, he's so blasé about it. He's like, whatever you want, man. Take all the photos, do whatever you want with them. They're yours. And so here we go. So here's the first, this over here in the top right-hand corner.
Sorry, Megan.
Shared. Sorry.
We're still on your first slide. I think you need to select your second slide. So just go down your slide.
I did, and it didn't work. Let's see here. Let's try this one more time. Is it gonna be picky? What about, can you see it now?
Yeah, we can see it now.
Okay. That was weird. Okay.
Megan, actually, when you click slideshow, your screen freezes. So
oh no.
Just don't, because I think you probably have two separate screens or something of the sort. So just don't press slideshow and it should work.
Okay. Sure. Don't press slideshow.
Yep, just scroll on through.
Okay, sounds good. So here you can see in the top right-hand corner the original photo. You can see the painting that I did over here in the top left-hand corner. Just for reference, I've included a lot of these original photos in with the paintings so you can see how they've translated and how my work has changed. I've done this in chronological order in a lot of regard, or for a lot of the photos are in chronological order. I think chronological order is really interesting for an artist because you can really see how their work has changed, what they've learned. You can see the tie back through it all. On the bottom of this one, you can see an abyssal grenadier shot that I did as well, also from Professor Jamieson. My work shifted pretty quickly from being a lot tighter and a lot more sculpted into more of this impressionism, which is what you see in the bottom, where you see a lot more brush strokes. From this work, I went into a series of drawings. Inktober is a month-long art prompt on Instagram, and it's all about drawing. It's about following the prompts, getting a drawing done, drawing every day. You see a huge skill increase when you do this as an artist, because what is practice for? Practice makes you better. So this was a series of drawings. This was also within the series, not all deep sea, but by the end of this, I had 31 drawings, and I thought, man, there's less than 31 letters in the alphabet. Maybe I should try to do a kid's book with some of these drawings. So I proposed them to a publisher that I had worked with before. I published a book of knitting patterns in 2020. Approached that publisher. They said, the drawing is too sophisticated for an ABC book. I said, well, thank you, but that doesn't really help me out here because I really want to publish a book. And he said, show me color. So we went back and forth a little bit and decided on the deep ocean as the topic. And so these are some of the finished paintings for The Unusual Ocean Who Lives in the Deep, which is releasing in June 2026. It covers 7 deep ocean habitats. It covers 19 organisms. And basically we start from the most shallow at about 1,000 meters and we go all the way down to about 11,000 meters. We cover different habitats. You can see here the submarine canyons with the beautiful pink bubblegum coral and the brittle star. Then we go into cold seeps, and we have the Bathymodiolus childresi mussels and the purple sea cucumber. We have the cold seep tube worms. Talk about cold seeps. We talk about hot vents with the giant tube worms and the scaly foot gastropod. And we have a whale fall with the hagfish and Ossidax worms. We go into the abyssal zone with Grumpeteuthis. There's a little submarine in there. The book is written for ages 9 to 12. It could be enjoyed by anyone, but that's sort of the reading level is that we're looking at. So that's coming out in 2026. And then what else do we do? What else we got here? Oh, we did a bunch of stuff with the book already. Dosey newsletter featured one of my paintings. I had an art show with a friend. This is a picture of Dr. Lindley, Dr. Thomas Lindley, and he is fact-checking my book on a boat in Antarctica. This is the coolest thing ever. And there's a photo to prove it. Other things I do with deep sea imagery, I like to take some of these deep sea images and I like to put a more personal slant on them. This one with the grenadiers, this photo, you can see it was taken in Antarctica over this past winter break for Antarctica's summer break. So December, January kind of time frame. The painting is to the left. And then I've added in these little symbols that are a blending of the otolith outlines. So this is an abyssal grenadier, Coryphaenoides armatus. These are the otoliths, and I've turned them into this little symbol that looks kind of like a butterfly. So I'm thinking about ephemera. I'm thinking about how transitory it can be. First of all, to see these animals it can be a very transitory experience. We cannot go visit them. We snap a photo. We have to deal with what we get in the photo. And then the fact that our oceans are warming and these animals need really cold water. Professor Jameson was saying it's one of his suppositions that perhaps this many in one area means they're breeding here, but we don't know what they're doing here. And so if we warm the ocean to the point where they can't go any colder because they're pretty much as cold as it gets here, what's that going to happen to them? That makes this a very transitory kind of experience. Up in the other side here, we have attachments, anxious and avoidant. Based on this deep sea starfish. I don't know the species. I have been sent the reports, but I believe this one was undefined. And we have a couple here with a deep sea anemone on the left, and you can see the reference photos that I use down here. And then this one on the right is called the people pleaser. And it's just sort of taking it a step further, using these deep sea holothurians as a basis for investigating ideas around interpersonal relationships that are a little bit more subjective for me. So there's sort of the two different things going on in my work where I'm trying to represent really beautifully and more accurately, and then also taking the work and making it a little more abstract and using it for my own subjective purposes. Coming up, this is a fun project. This links into our corals, Paola. So I'm working with a researcher out of Sweden. Her name is Laura Beckman, and I'm working with two other artists, one out of Juneau named Kirsten Keller and one out of Corvallis, Oregon named Nilanjana Das. And we are doing a giant project based on footage of deep ocean cold water corals taken off the coast of Alaska in 2023. So I've included the QR code. If you want to watch the video, snap it up, baby. Check it out. It's really cool. There's a lot of hours of images, but like, I'm into it. I'm there. If I'm there, maybe you could be there. And this is a painting in progress. This is the Uliaga seamount. I think I'm saying it correctly. Uliaga seamount, definitely in progress. This particular painting is booked to go to Alaska in February. We have a show at the Juneau Arts and Cultural Center there. And then we have a big project that we're trying to put together in Spokane for April of 2026, an art show, field trips, public lectures, school lectures. We're flying Laura over. We've applied for a big grant. We received a big grant from the British Ecological Society for that already. And so we're still in the putting all the pieces together phase, but I had to start on the art. And then finally, where are we at? Is my socials, if you're interested in checking them all out. Website, Instagram, and Blue Sky as well. So I think the one thing I forgot to say that I wanted to mention was I work within the art movement of Impressionism, modern Impressionism. Impressionism is an art movement that puts a lot of emphasis on light and how light moves, how light changes. You've probably seen Monet watercolors. You've probably seen like 15 of them. And you're like, why is this man painting 15 water lilies? Should have said water lilies. 15 paintings of water lilies, and they're all different colors. Why is he doing this? Because he was observing the light. So he was painting them at 6 o'clock, 7 o'clock, 8 o'clock, 9 o'clock. He was painting them morning. He was painting them midday. He was painting them evening. It was very scientific in a way, where he's trying to record how the light moves, how it changes, which colors come to the foreground, which colors recede, looking at diffusion in the atmosphere. How can that be represented within Impressionism, which is these really chunky brush strokes, a little more subjective color. You can get really playful in there. A little more abstraction. It's not really smooth. It's not really graded. So I personally think that because we can only view the deep ocean with these really harsh lights, right? We can't bring the deep ocean to the sunshine, and we can't bring the sunshine to the deep ocean. So we only see it in this really harsh way. But I think that's fascinating. I think it really can illuminate some of these adaptations that these animals have. For example, the deep sea fish, the snail fish, the grenadiers, they have these pockets of jelly within their flesh. Okay. And it's because it's easy to create. They can grow really fast. They make this jelly, it's like cheap to synthesize in the body. But what does it do? Because when I'm looking at these photos, and maybe we can go back here. When I'm looking at these photos, some of them have like a haze around them, or a blue light around them, or this one at the top with the supergiant amphipods, how they reflect colors. I mean, these animals are intrinsically white, but they're kind of transparent, and they're really reflective, and they're showing different colors, but they have a little color in and of themselves. And those are the things that I'm really fascinated about with these animals, is the lighting, how their adaptations and their scales and their skin and their jelly interact with the light. And then how can I convey that to the viewer in a way that promotes curiosity? And they can walk in and they can see an item, they can see a painting that's really beautiful to begin with, because so much of our perception of the deep ocean is, well, that's weird. Oh, that's creepy. Oh, that's freaky. I don't want to be around it. But they walk in and they see something beautiful, and they're like, what is that? So they're engaged and they're curious. And then maybe they read the title plate and they're like, oh my God, I had no idea. Like, I didn't know that there was a beautiful pink coral down there. That's so fascinating. So I guess that's maybe my mission statement right there. And I think I'm done. Does anybody have questions? We want to chat it up. What are we doing now?
Yes, it said if anybody has any questions, feel free to pop in for Paola or for Megan. No question is out of the table, you guys.
I mean, I have a question.
I should stop sharing, I think. Yeah.
Question that comes up as you were talking about all of these beings. What keeps coming to mind is like the speed that things happen in the deep sea. So Paola, you were mentioning like gigantism, but I'm curious if either of you can talk about like how fast does life exist there in the deep, cold, limited oxygen space? I have some assumptions, but yeah, curious if there's more to learn about that aspect.
Yeah, no, that's a great question. Megan, do you want to chime in, or should I start?
You go ahead. I mean, I think you probably have a little more meat behind your answer, because mine's going to be like, yeah, they live a long time.
Yeah, it takes a long time. Precisely that. That's a great question, John. At least for the deep sea, but especially when it comes to minerals, they can take it's been are in the news around a lot when it comes, for example, for these ferromanganese noodles. And then we learned that when we look at these ecosystems, up to 95% of the organisms of the entire biodiversity that you find around them live on them, it takes millions of years in the first place to form these ecosystems. And then for life to be able to colonize them, that's a whole other thing. So things in the deep sea move relatively slowly, especially when we look into the bentos. That when we talk about the bentos, we talk about the seafloor. And then scientists will usually, like, if someone concentrates more on studying the seafloor versus the pelagic region, that's a good distinction to make. I think things at the pelagic region moves on a different pace. But yeah, all in all, it moves really slow. It takes a long time to form, and they can also live hundreds and hundreds of years, especially if you're looking at this, like, I just love Meghan's painting, so I'm gonna keep pointing at it, the lamellibrigia worms that she has on her background. So yeah, that's in terms of life when it goes to that, because then you also have these really cool, and this is a long one, but biogeochemical cycles, just anything that you can think around that goes around, like the carbon cycle, the nitrogen cycle. We love to talk about the carbon cycle because it connects right up to our atmosphere, especially when we talk about greenhouse gases and climate change. It becomes important. The deep sea is a massive carbon sink, and it is essential when it comes to managing the balance when it comes to the absorbance of carbon dioxide from the atmosphere. So that's a whole other topic, but that's also, it moves at a different pace, and it's a delicate balance as well. So there are different timelines when it comes to the deep sea, but when it comes to life, like, it can easily be hundreds of years. And it's really cool that when we go with an ROV, like a remotely operated vehicle, and we like Meghan said, we shine light, they don't see light in the first place. We might be the only ones, and then they're gonna tell their friends, they're gonna be like, oh my God, there was this alien and it shined light on us. And they're gonna think they're crazy because they don't even use light. They don't need it. So I love Meghan's perspective when it comes to using colors, because we're shining white light into it, but they don't use light. They don't need it. They mostly depend on bioluminescence, if anything, when it comes to light. So yeah, different timelines, and we only see this tiny snapshot when we see these giant worms or these entire, like, massive ecosystems, and we think like, wow, this is huge. It must have, like, and it does have a lot of activity, and some timelines, like, some cycles move faster than others, but in the main thing about the deep sea is slow. And when we talk about slow, it's like really slow, like sometimes geological timeline is slow. But Meghan, what do you think on that point? Like, how do you capture those ecosystems, right? If they move, when I think about art, like, it must take a lot of skills to be able to capture what's happening, like, in the entire deep sea in just one snapshot. Like, this is not something you can come back to and say, like, hey, like, I need to get this side of the organism, right?
Well, yeah, I mean, you're really restricted to the photos that you get, right? And so I'm really lucky that I do have access to a lot of photos. Professor Jameson has been very generous. He sent me a huge amount of photos, but you have to really comb through them. Sometimes I actually will often combine them in Photoshop if I'm doing several different angles and stuff, making sure it's the same species, making sure it's all relevant, and then put them together and then paint from them. I don't know, I think it's fascinating that it's like a snapshot in this incredibly long life, and some of these animals are going to live longer than I do, and I think I feel really honored, like really, really honored to be able to access the imagery and represent them and, like, paint their portrait. They'll never know, but they have a beautiful portrait painted of them, and I think I don't know, there's something really fun in there too.
Meghan, there's a question in the chat, since you're on right now, that says, you've been talking a lot with these scientific names and a lot of information about the organisms that you paint. Do you read the literature directly, or are you exploring more science communication from DOSI or the podcasts, etc.?
I read a ton of papers. All of my research that I did for the book was all straight from papers. I give a huge shout out to Dr. Lindley, because when I was looking into and, like, researching, putting this book together, I reached out to him and I was like, hey, so how does this work? Like, what's the best practice? Because I'm a how it works kind of person, right? So I'm like, what's best practice? How's it work? And he said, follow the research until you can't follow it anymore. You got to find the primary source of that research. And so I was like, all right, I can do that. And so I do read a ton of papers. I ask a lot of questions. I feel like if you're friendly and kind and you say please and thank you a lot, people are usually very excited and happy to share their research with you. A great example, on Blue Sky one day, I had posted that picture of the starfish painting, and someone had commented on it and said, oh, I researched starfish and I just released this paper. And I was like, oh, where is it? What's it called? And he goes, I'll send it to you. Sent me this 20 page long PDF paper. And I was like, this is amazing. So people are really, really excited to connect when you find the thing they're interested in. So yeah, Ziggy, hi Ziggy, it's super nice to see you and or hear you or read you. And I do research papers as far along as I can get. I try to read the Dosey newsletter. It all, I'm just obsessed. It's all just like little nature facts, and I need to eat them to survive.
And for whoever wants to answer as well, is do organisms rely on bioluminescence to communicate or to mate, or what is the reason for it?
Yeah, more than happy to expand on that one. And on Mega's point, when it comes to scientists, something I've learned, like they love to talk about their science. Like we're like little kids. So if you like send an email to one about like, hey, I'm doing, especially if you're doing art around their science, oh, it's like they're gonna be more than happy to take a coffee break and talk to you about it. So the usual, like the typical scientists, they are very open and they love to share their science. There's nothing gatekeeping, like we want to get the word out of these amazing ecosystems. So just as Meghan said, like feel free to ask questions, get those emails, send them out. They're gonna be really happy to respond usually, or if not, maybe the second or third one that you reach out to. And she also mentioned Dosey briefly, the Deep Ocean Stewardship Initiative. That's an amazing resource that you can use. We are an amazing group of, they has researchers, policymakers, and they all come together to just share the world and these deep sea ecosystems in the US. So also another great resource. And yeah, when it comes to bioluminescence, yes, it is mainly, it depends on the organism, and it can be mostly to communicate. And it doesn't come to my mind about any organism that uses it for mating. I wouldn't think why, I don't see a reason why not to. I just want to say yes, of course, and then nothing comes to mind when I think of one. But it is, I know for sure that yes, it is mainly for communication. They are really smart on that. So cool.
And aren't there, okay, so correct me if I'm wrong. Sometimes I conflate things together, but I thought there was a kind of squid or maybe shrimp that could like let out bioluminescence and then run away. Can't it do that? Can't it like shed a shell of bioluminescence and then run away so that the predator thinks it's staying in the spot where it's got sloughed off the shell?
I think there's a lot of different organisms that do it. Like there are like the vampire squid. It just like shallow depth squids, they release ink to kind of obscure themselves, or some ink even like it's mimicking the shape of an octopus. So it's like a mucus cloud filled with this dark ink. But vampire squid, they release bioluminescent ink or this bioluminescent mucus. So they do that and then they dive to the side. But also this is like super common in a lot of other types of organisms. I do a lot of polychaete research. So there's, I forget the name of it, but I think it's like the bomber worm or something, and it just drops like these green flashbang bombs to kind of distract whatever's hunting them. And with bioluminescence as well, I mean, like it's like talking, you can use it for so many different things. You can use it to mate, you can use it to find friends, you can use it to talk your way out of a difficult situation. Bioluminescence is a lot like that. There's like some, the anglerfish, it uses this little lure at the top of its head to kind of fool other organisms to thinking that it's an organism that wants to mate. But I think a common trend, at least with bioluminescence, is that it costs energy. So there's got to be a reason to do it, because it's like you're always in an energy deficit in the deep sea.
Well, and so many of these really cool, I mean, what I would call like deep sea superpowers, they do have a cost. They have an energy cost, like when the hagfish slimes and it releases these slime cells, and it takes a long time to recoup that and to rebuild them, to use them again.
Mm.
Christian, can you tell us again what you study? Because I kind of missed that.
Yeah, sorry. So I'm a PhD student at the University of Bergen. I'm working with hydrothermal vents and what's it called? Cold seeps. So just like there are these siboglinid worms like the Lamellibrachia, the Riftia that Megan's beautifully painted, there are these lesser known frenulates. So these ones, they're the big famous ones, but frenulates, they're like smaller, more hair-like kinds, and they've only been described in the Arctic in kind of what we call a qualitative way. So, we'll say these are big forests, these are small forests, but how big, how small are they? And so my PhD thesis is set to describe the variety of worm forests in the Arctic. So how do they vary based off methane seeps? How do they vary based off hydrothermal vents? And which animals like which kind of forest? It's kind of like also we're trying to relate them to land-based forests because there's this emerging concept of animal forests, a super great book by an author, Sergio Rossi, trying to introduce like even shallow water coral reefs, calling them animal forests, just because this whole idea of them forming a habitat is very similar to how trees provide habitat in their forests. So just making it a bit more accessible. And I've found that when I talk to people, I've worked a lot in science communication too, that they have an easier time understanding it. If you tell them, well, there are different kinds of worms, just like there are different kinds of trees in a forest, and some of the same species of trees grow tall, others grow short, and some grow very densely, and so on. And that leads to different types of animals living there in different amounts. So we're trying to do that and then relating it to like protection against deep sea mining and stuff since it's Norway and so on. So that's a little bit of what I'm doing. And then also I'm in the Deep Sea Podcast Discord with Megan. So that's how I found out about this. So I just wanted to hop in and support my buddy.
Thank you. I want to go back to methane seeps because I know that Paola has worked on methane seeps in Antarctic, and I have an enduring love affair with Antarctica, and I really want my next book to be on Antarctica. So tell me everything you know, please.
Oh my God, yes, I'm on the spot because my colleague that's more senior than I am is here as well. But yeah, when it comes to Antarctica, as you know, it's very cold. It is kind of dark, half of the year. We only get one sunrise and one sunset. So it's more like it's kind of a parallel narrative to the deep sea more than we think. And our lab, the Cold Dark Ventos lab in 2011, discovered the first ever methane seep in Antarctica since the moment it started. And this was because it was by accident, as most scientific discoveries are. They have been diving there since the 60s and almost each year. So in 2011, when they went diving, they noticed a microbial mat. And one thing about methane seeps is that you can see, for those who don't know, like we know the carbon cycle, these are just bubbles of methane, like gas methane coming out. And you can have them like a beautiful stream of bubbles coming out, or you can literally have nothing, but then you put a sensor and you know there's an increase in methane there. So by some shape or form, there's a methane seep going on there. And they're pretty common in our oceans. That's the first thing, but not one methane seep has been described in Antarctica up until then. And they noticed it because they noticed a microbial mat. And this is because, well, you have methane are carbs, it's like bread to us, and microbes love that. So they're one of the first ones to react when it comes to having this influx of methane, of nutrients, in a region where there are not a lot of nutrients going on. So we have this, what we call a biofilter, because they get this methane and eat it up, and then in turn they produce carbon dioxide. So we have a really, really bad greenhouse gas to a less bad greenhouse gas. So we like that. So they noticed this in 2011, and since then the lab has been going there not every year, but more or less since then, to study the evolution of this methane seep, since we have the rare chance of being able to visit this site more than once and since the beginning. So we're studying from its microbial succession all the way to when it's starting to influence meiofauna, which is the organisms that live in the sediment, to the ones that live on top of the sediment, and then to even potentially sponges and megafauna, stuff we can actually see. That's what I concentrate most on, the stuff that's above the sediment. So yeah, that's the main overview when it comes to the methane seep. We've been studying this since 2011. We have a lot of data. When it comes to art, Omega, I would love to talk more later on that, because we actually do bring like an artist with us like each year, and that's something we do. That's more of my advisor's realm, but we're more than happy to make the connection. And yeah, that's usually how we do, because the same thing that you're doing, when it comes to the deep sea, most of the people don't know this ecosystem exists, and science can only get us so far. We're bringing the knowledge out there. We need people to share it in a way that people relate to it and make a connection with it. It's not like we can think of the moon that it's remote, but people see it every day, right? So NASA has an easier job than NOAA in the US when we think about it, when it comes to making people relate and connect with these ecosystems or systems in the moon. So yeah, that's when it comes to Antarctica. That's where we're working on right now. My colleague Jacob is here. He works with the microbes and all the different metabolisms that go around them. Like, how are they eating up this methane? Who is eating it? Like Antarctica, like most of the stuff that lives in Antarctica is only in Antarctica. Like, we can find some of the stuff that it has cousins maybe around in Chile and Argentina, like those southern places, but because of the polar front, this uninterrupted current that goes around Antarctica, most of the stuff there is being described for the first time. And when it comes to microbes, that's a whole other realm that we're only starting to scratch the surface on what's happening there, especially with this new methane seep that's introducing methane there. And Antarctica, it's estimated to have up to 75% of the marine methane reservoirs. So we really need to know what's going on so we can do proper estimates and how can those ecosystems look like in the future. And especially when it comes to climate change, right? We want to know what's happening before the change starts happening, if it hasn't already. That was a lot of information and I talk really fast. So
I was totally here for it. Totally.
Oh,
I did see a question from Jacob on the chat. Have you spent time on organisms with bioluminescence, and how do your methods change when the light is coming from the organism itself? Okay, so the first thing you need to know about representing a photograph as a painting, don't think about what it is. Look at what it is. And if you need to turn it upside down so that your brain can't recognize what you think it is, do that. Because what you're really trying to do is duplicate shapes and colors from one space to another. And if you're thinking, well, this is bioluminescence, so what it's going to do is turn your left brain on, and your left brain is all about language, and it's all about being verbal, and it's all about making those very concrete connections. And what you want to do when you're creating art is turn on your right brain, and your right brain is colors and it's shapes. And it's very quiet, and it's very happy to turn off, because your left brain's really happy to turn on, unless you're left-handed, and then your balance is a little bit different. So my answer is, representing something with bioluminescence, I have done it before, and you're really just looking at the shapes and colors, and you're just breaking it down. If you think about it like a coloring book, and if you think about, okay, I'm looking at this piece of paper, and in this area there is this shape that is this color, and then you just try to replicate it as best you can, and that's going to be probably the most successful way. There's a lot more going on with layers, glazes and stuff that I do as well. But like, the basic concept is you're trying to replicate one shape and one color from this one to this one. And don't think about it. Don't think I'm drawing a tentacle, or I'm drawing a suction cup, or I'm drawing a head fin. Don't think about that. You're just looking at the shape to the shape. So that, I don't know, I hope that answers your question.
I would encourage everybody to go look at the ROV videos and get a snapshot, and then try drawing just like Meghan did. I feel like everybody would really enjoy that.
And I was super serious when I said practice, like practice. You would never pick up a violin and go play a concert with an orchestra, having never practiced it. Why would you try to paint a giant painting having never practiced it, and then expect to be painting at the same level as someone who paints every day for 4 or 5 hours, right? I would never walk into a lab and expect to know how to use everything in the lab, because I didn't have any practice or experience. So just practice, 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 and don't stop.
Okay, I'm seeing another question from John. That methane isn't even more of a potent greenhouse gas than CO2. Yes, but at least 34 times more potent. Do we know how much of the methane makes it to the surface from the seeps, or have you uncovered tools from your research that could impact the current situation above waters? That is a great question. I think it depends on the seep stage. Like right now, what we're studying in Antarctica, it's called the cinder cone seep in McMurdo Station. I would call it like a baby seep. Like it's still like a seep to be able to be fully functioning ecosystem where even the megafauna, it can have symbionts and just organisms that benefit directly from the seepage by having microorganisms inside them that are able to synthesize these things. It takes hundreds of years. And our seep is maybe 15 years old. So it's a baby seep. It's just starting out. We know the microbes for sure are interacting with the seepage. Jacob is uncovering those metabolisms. So when microbes are there, depending who is there and how much of them are there, is how much of that methane is not going to the atmosphere and instead is being recycled into the ecosystem and turned into something else. So that would be the first thing, that it depends on the seepage and how mature the seep is. And like I said, seepage is very common in our oceans, especially around, I think, continental slopes. Like they're everywhere, and they're mostly on the deep sea, but they can be as shallow as 10 feet. Like the one in Antarctica is only, Jacob, please correct me, but I think it's 7 meters deep. Like we actually go scuba diving. We don't use any videos. We do make a hole. They make a hole for us in the sea ice, because the ocean is the frost sea is a bit frozen at that moment. And then we go scuba diving through that hole in the ice, and I directly study the seepage type, which is also a very rare opportunity and a really cool place to do experiments when these ecosystems are actually usually just very, very deep. So when it comes to a rate and having a rate of how much methane goes into the atmosphere from these seepage sites, we truly do not know. I think we can do some sort of correlation with the methane concentration that we see at our seep, and that can definitely be a starting point, but I don't think there's just one big number that we have that this is the amount methane seeps are. It might change with climate change too, not in a good way, but yeah, that's a great question that I cannot answer right now, but definitely something I think some scientists are trying to work out, especially when it comes to modeling. Hopefully I sort of answered your question, John. Okay.
Yeah, I'm just kind of curious about like the filtering process. Like these creatures are consuming some of the methane or the energy that's available there. So yeah, it's just how do they do that? It's just a foreign process compared to what I'm used to with trees and like just the oxygen life forms on the surface.
That's a great question. Antarctica is the very rare instance that we're gonna have seepage and also a lot of oxygen going around. It's very well oxygenated, those waters, at least where we are. And though saying that, which has nothing to do with what I'm going to explain right now, it was an intrusive thought when it comes to Antarctica, is that the question was around the biofilter and the microbial mats. Yeah. So there are different metabolisms that go around, and when the easiest way to think about it is that we know plants do photosynthesis. They have these structures that are able to accept these photons, and they have this electron acceptor that works around and do its magic and then produces oxygen and then energy. When it comes to these chemosynthetic communities, where what happens is that instead of using Jacob actually, this is his study, and I don't like relaying questions, but he's the one that actually studying metabolisms when it comes to these microbes that we have. I study mostly the sea stars that eat the microbes. So Jacob, can you expand on chemosynthesis, especially with the microbes that we're seeing in Antarctica? I know they're just weird, so I don't want to lie here about how it goes with the microbes there. Like, how do they eat up this methane?
Yeah, absolutely. So the methane is oxidized to carbon dioxide, and that can be done in a number of different processes. So it's essentially used as a component of respiration. So
where you would use, especially in anaerobic settings, where you would use oxygen, these organisms are actually using methane as that electron acceptor. And this reaction is kind of on this like knife's edge of energetic favorability. So it's like you're barely getting any energy from methane respiration, which makes it a pretty interesting metabolism to study, because this methane filter that Paola was talking about is established extremely slowly. It's so energetically unfavorable that it takes 7 months for a single anaerobic methanotroph to divide. And to actually start filtering methane, it takes on the order of decades. So one of our big questions is when is this methane filter going to establish on a new seep? But there's a whole bunch of different metabolisms. Some are driven by methane coupled to sulfate reducing bacteria, and some to nitrate reduction. And that is what we found in Antarctica, which actually is more energetically favorable and responds faster. So it seems like the community is kind of building that methane filter faster in Antarctica.
Thanks, Jacob.
Yeah.
Would it be okay to just chime in with a quick follow-up question? Because I've always explained the concept of chemosynthesis in general, just like as very similar to photosynthesis, the only difference being, or the main difference being, that photosynthesis is driven by light and solar energy, while chemosynthesis is more driven by what did you call it? Like the energy from chemical potential. So like you have sulfides in contact with oxygen. Would that be a fair comparison?
Yeah, I think we just mainly changed the electron acceptor.
Mm, mm.
Can you expand on that, Jacob? Since it's a micro metabolism question, this is so out of my field, so I really appreciate it if Jacob chiming in, because
No, I'm also a fauna person because I just want to get educated here too.
we can talk about polykits, so I love this. Yeah,
Good
go
stuff.
ahead, Jacob.
So chemosynthesis, it's kind of a broad term, a blanket term, but chemosynthesis is partially where you're respiring things other than oxygen, and you're also gaining carbon from usually kind of stranger molecules. So in methane oxidation, you can sometimes be assimilating that carbon into your body as a microbe. And that is something that Paola studies, where that carbon assimilation, you can actually trace up the food web, and you can figure out how much energy is coming from the chemosynthesis versus the versus like photic
Yeah.
carbon assimilation.
So some of the methanotrophic bacteria, they get their carbon directly from the methane and not like inorganic carbon dioxide like most photosynthesizing organisms would do.
Yes.
Okay, dang, that's pretty cool. Thanks.
Thanks, Jacob. Yeah, what Jacob was mentioning is that one of the techniques that we use at this lab, we use stable isotope, which just means that we have different types of carbons with different number of electrons, and they change, right? And so depending on what type of food you eat, basically. So if this organism eats something that has the microbes that are signature, so this is just a value, and I compare those values, the more negative they are, potentially just means this is something we can broadly expand really quickly, and this can be hours, so I'm going to keep it very short. But basically, the more negative these isotopes are, the more indicative it is that the diet of this organism is coming from methanotrophic related food web. And this is something very, very relative. It depends on the system that you're looking at and where you are at, and you usually want to measure the signatures of both any photosynthetic source and any chemosynthetic source, and then compare those. And also another thing that we use here is fatty acid analysis. Something about fat, especially us too. When we eat something fatty, it doesn't really change the composition if we don't digest it. And these microbes, they produce some really unique fats. So when these big organisms go around and eat these microbes, or they're like scavengers and eat some of it around, we can, if they ate that, some of the fat that this microbe produce, if I take a tissue sample from this sea star that is going around, I'm able to see if this sea star benefited or at least ate these microbes. So I can connect this megafauna community to this methane seep community. And that's how I can like start linking up. Are they dependent on the seep? Are they benefiting? Do they care at all that the seep is there? So far, or like I mentioned, our seep is a baby seep. The data so far is showing that they don't really care, like if they eat here or they eat there. But this is a really interesting result because this is the first time we're actually thinking like this when it comes to methane seep in Antarctica. So yeah, all cool things that we do to try to understand how food web works. And the more this methane is recycled in the system, is methane that we're not seeing in the atmosphere. So we like that.
So cool.
So guess what, you guys? We're having lots of fun, but we're getting down to our last minutes of our meeting. John, do you want to chime in?
Yeah, I just wanted to appreciate this conversation because this was definitely not where I expected it to go, and I learned things that I didn't know were possible to even consider. So just really appreciating that. And wanted to just do a quick share that Paola is one of the Ocean Leaders Fellows with Sustainable Ocean Alliance. We're building the largest network of young ocean leaders around the world. We have a few of the other fellows in the audience as well. And I wanted to share that we had created a Deep Sea Academy. So this is shared on our community platform. And this is an opportunity to learn more about what's going on with the campaign against seabed mining, and just really to get an additional sense from a political perspective. And it's about, there's 4 different modules to the program, definitely worth checking out. And also wanted to invite you all to the next one of these. Rita, when is the next one of these? And what's the topic?
It is August 15th, and the topic is exploration. And so it'll obviously depend a little bit on our both science and our artists, just like Paola and Meghan with their disciplines. But we will be having the Reef Chats once a month. Right now, I think we potentially have some that cross over into the next year because there's lots of different topics that you guys are interested in, and we want to be able to have these amazing talks and not only learn from our wonderful speakers, but also from like people that join us, like Christian and Jacob. So thank you so much. We want to thank everybody for being here and taking a chance, and invite your friends over, because an hour seems like a very long time, but not when you're having fun.
Awesome.
I added the link to the SOA article that talks about the Academy. If you do not have an account, I highly encourage you to create one. You can connect with wonderful people there. And we will be posting the video also on YouTube. Any links that we found that are useful, Paola and Meghan will also be there. And if anybody wants to share it and rewatch it again. So thank you everyone for being here. And I hope to see you on the next one.
Thanks, y'all. If you're open to turning on your microphones and saying bye, good to see you. I'll see you next time.
Thank you.
Thank
Thanks
you
for coming,
all.
y'all.
Thank you
Yeah.
all.
Bye-bye.
Thank you.
Thanks. Bye.